All right, does it work? Yes. Right, welcome everybody to this camera event. This is a keynote speaker event, and today we've got two fantastic speakers from Pavia, Italy. Surprising, given the fact I'm Italian, isn't it? But I'm truly excited because I think today you're going to see something that is very difficult to find in research. So trying to model something that we don't know much about, like the brain. And Egidio and Claudia will try to show you how they integrated experimental and modular approaches to do that. And something about the two speakers. So I'll start with Egidio, who's going to start first. Egidio is a full professor of physiology and co-chair of the Department of Brain and Behavioral Sciences, and also director of the Brain Connectivity Center in Italy, or the IRCCS Mondino. Its main scientific interest is about the cellular and circuit functions of the cerebellum and its pathology in the context of the whole brain activity. Well, I could talk about Egidio for a long time, but so I know that I got a first grant, actually ERC grant, so European grant, many years ago. And the thing is that it never stopped getting ERC grants. So that's I think it's a good thing to say. In terms of the second speaker, Claudia. Claudia, first of all, is a great friend, but also an excellent bioengineer. And she was much better than me when we studied together. Without her, probably I wouldn't have finished my degree. But apart from that, Claudia studied at Politecnico and worked at the Politecnico of Milan as a neuroengineering, neurorobotics, and rehabilitation in motor learning. And then she moved to Pavia, and she's now coordinating the neurocomputational lab in Pavia. Both Egidio and Claudia are working on the human brain project, and I think they are better placed than me to talk about it. So without any further ado, I pass over to you guys.
Thank you, Dario. So thank you, Dario, for the invitation. I will be the first to start, so I'm very happy to be here. And I will try to make a complex story simple to grasp. So my job will be to make it simple. Claudia will make the job of making it more complicated, yeah? So what I'm going to talk about is how the brain can be modeled. Well, you can say, well, there are many ways to model the brain, sure. In the last years, we have followed this issue from different point of view. And you will see that there are many ways to model the brain, and the way that we have favored is to start from bottom and to climb up, at least in some phases. So we start from the knowledge that we have on neurons, on microcircuits, on the cells of the central nervous system, and then we reconstruct microcircuits. Microcircuits that work as if they were real. This is the reason why we call this kind of modeling realistic. The other way of modeling the brain is to come from top and going down as a top-down approach. The top-down approach is favored when you are facing a complex system with ensemble measurements of the system activity, and you don't know very much about the elements that are generating this activity, and so you're facing an inverse problem. I will come into this very shortly. All this activity has been developed in the last years into the Human Brain Project. It's a European project that lasting for 10 years, will finish next year into 2023, during which we have contributed to develop what you will see, plus technologies to generate the models and to run the models in an easily accessible manner. Okay, so there is also an infrastructural activity. When we study the brain, we have to face a couple of issues. And I think the three main issues can be summarized in this way. When a certain part of the brain is activated. It's very important because the brain has a topological architecture, in which the different parts carry out different jobs. And therefore, a first and very important issue is to understand when a certain part of the brain works. The second point is what the brain computes, or what a specific part of the brain is computing, because the brain is a machine, basically, a physical machine. And this physical machine generates computations. And this computation is not easy to understand. We have to understand what the circuits are doing. The third issue is how the circuit do that, because we may be happy to understand what the circuit is doing, but without any kind of knowledge about how the neurons are doing it. So we would like to understand how the neurons are carrying out the job. And this is why in some phases of our work, we start from bottom, we start from the neurons, and we climb up. It's very difficult to infer what the neurons are doing by taking an ensemble measurement. It is easier to understand what they're doing starting from the neuron themselves and going upward. Finally, there are implications for medicine and for ICT and for artificial intelligence and all the correlated fields that descend from the fact that we understand how the system works. So we want to have a sort of causal understanding of the system, not a superficial understanding of the system. Okay. Now, a crucial point is the multiscale organization of the brain. So when we interrogate the brain, we needed to interrogate multiple levels, not one level that explains everything. Suppose that we want to understand what happens at the ensemble level, which means that the whole system, we can use techniques like magnetic resonance imaging, electroencephalography. We can take out ensemble measurements. We don't understand much about the neurons, but we understand something about the ensemble. If we go down a bit, we can take measurements from multiple neurons, and then we have an understanding of how the network operates. Or a bit better, I would say, how the different neurons in the network interact. If we go down more, we can go to the single neuron level. Here we can understand a lot about the single neuron, but the perspective of neuronal integration is very, very far. So we don't understand how this neuron will integrate with others. So we have to complement all these points of view, going bottom up and top down. Okay. Now, this is an example of the top level. Since I will speak mostly about the cerebellum, I show you more pictures taken from the study on the cerebellum. This is a reconstruction that was done in our laboratory in collaboration with the field at UCL, the ION at UCL, in which there is a reconstruction of the tracts connecting the cerebellum to the cerebral cortex. The point here is that the cerebellum is strongly and heavily connected with the rest of the brain, in particular the cerebral cortex through the thalamus, with millions of fibers. And these fibers do not go only to motor areas, but also to cognitive areas and associative areas, which is a sort of change of point of view. Because the cerebellum is classically considered as a motor organ, actually is a motor organ, but performs a cognitive operation, which complicates a bit the story, but for other reasons make it a bit easier, because it means that the cerebellum uses a generalized algorithm that is applied to every kind of computation. So our aim now is to understand the general algorithm of the cerebellum. This is an example in which the cerebellum is activated during the execution of a motor action, for example, squeezing a ball, like here, or is activated by observing other people squeezing the ball, which means it's not involved in directing motor actions, but in imitating, understanding, interpreting, predicting what the others are doing. It's a short part of a mirror system. You know, the mirror system that is considered to be so important for our cognitive functions. The cerebellum takes part to it. This is another example in which the cerebellum is involved in what we call the resting state networks. Please, if there is something that you do not know, please stop me and ask before I go forward. The resting state network are assemblies of networks in the brain that pulsate synchronously. So this kind of picture is the instantiation of synchronicity between areas. Okay? The areas that are colored in a sort, in some panels, for example, up here, here, it means this area pulsates synchronously. These are images of coherence. The cerebellum is involved in the coherence of the default mode network. This is the network for the internal the control of the internal state of the brain, which is very far from motion. It's something that is really also far from cognition. Is even more. It's the internal state of the brain. Is involved in the attention network, which means the network is switching from the internal reference state to the external reference state. So if you think about your stories and then at some point your attention is distracted, the cerebellum takes part to this change of orientation of the brain state. The cerebellum is also involved in the salience network, which is the network that controls the content of the images that come from outside, whatever they are. As in the example that we have seen before, we see the squeeze ball test. Somebody squeezing the ball, the cerebellum is transiting from the internal state to the external state of reference and moves through all these networks, changing the state of activity of the brain. Clearly, from all these examples, the message, the take-home message is, if we look at what the cerebellum is doing, if we look for what the cerebellum is doing, we have to consider an expanding set of observations, not only motion. Okay. Now, we can try to go more on the relationship between the neurons that are working in the cerebellum and the signal that can be observed during certain tasks. And this raises this kind of issue. If we know the signals, or the elementary causes, as we call them, we can directly generate the observed data if we have a good model in between. Suppose that we have a good model, that, as those that Claudia will show you. If we have a good model, we can start with the data, generating the signals, and then move up to the ensemble that we can measure in a real brain. The reverse problem is if we start from the ensemble and we go down to the signal. This is an inverse problem mathematically, very nasty. Sometimes it's not solvable in principle, but something we can do. You will see that we can do something on this problem and extract some of the information, not all the information, because the system is redundant. The system has a lot of alternative states. We can't solve them during resolving the inverse problem, but we can extract parameters that are related to the relationship between the ensemble signals and the elementary causes. So this is the world where I will try to bring you in during this talk. Okay? During these years of the Human Brain Project, we have worked quite a lot trying to understand how to go into the issue. And we have generated a kind of graph that you see here. In green, you have biological observations. All the models are data-driven because all the models that I will talk about are generated starting from biological observations. In general, they are data-driven. If you take the white box, the models here are bottom-up. They start from molecular properties, neuronal electrophysiology, neuronal morphology, microcircuit connectomics, to build detailed neuron models, detailed network models, and downstream of this, through operations of simplification that are mathematical operations, Claudia will tell something on this, I think. Through mathematical simplification, we can go to simplified models that maintain the same fundamental properties of the complex model. To tell you in simple words, a complex model may be done by 100,000 differential equations. I'm not joking. It's very big. A simplified model can be done by tens of differential equations. So it is still a dynamic system, but it is compressed in a way that we can maintain the fundamental dynamic properties with a small size representation. And with the small scale representation, we can grow up in scale with the number of neurons, so we can expand the network. We can use it more fastly, run it on computers, that can be efficiently solve the mathematical integration of the differential equations. And finally, we can use it for very interesting simulations. For example, the virtual brains and the robotic controllers that I just indicated in the title. Okay. The gray area is the top-down flow. The top-down flow tends to resolve the inverse problem and generating virtual brain models. There is an intersection between the bottom-up and the top-down flows here because we can use the models that have been constructed based on biology, and we put them into the virtual brains, or analogously into the controllers of the robots. So we are in this point. Basically, we generate a good model that is generated top-down, and we use a bottom-up, and we used it in the top-down flow. To come to this strategy, we have taken a couple of years. Discussions, problems, controversies, clashes, but finally this works. You will see something that I think is a good solution to this issue. Okay. This is an example of, let's try to run it. Wait. Aspetta, bisogna togliere. E questo è il mouse. Ok. Togliamo questo. Okay, now it works. So the one on the left is a realistic model of a neuron. It's a neuron of the cerebellum. You should be able to see the flashes, the light flashes into the neuron. And also simultaneously you see an electrophysiological recording simulated that runs. So the neuron is receiving inputs, is a Golgi neuron of the cerebellar network. It is receiving inputs and is generating outputs in terms of membrane potential changes, action potentials, and all that you know about the neuronal physiology. To run this kind of neuron, we are using about 25,000 differential equations. It's made of more than 1,000 electrical compartments, each one containing several types of ionic channels. And it's a realistic reconstruction of what the neuron is. This kind of neuron has been embedded into a network. The network is made up, in this case, of more than 10,000 neurons. They're of different types, not only this one, also other types. They're connected together. And at this moment, they're activated in background, but at some point you will see a green light here, and there is an input coming into the network, imitating the input from the peripheral sensory system, and then the neurons begin to work and to react and to respond and to generate some interesting dynamics that we are investigating. Now, all the technology and the principles behind this will be explained by Claudia. I will go through some examples of this I also skip for the moment. I go through some examples that show you how these models are used to represent and then understand the physical system generated by neurons and synapses. The one on the left here is a representation of an experiment. These are experimental recordings in a configuration called the voltage clamp. These recordings show the currents generated by the neuron when it is stimulated by voltage steps. So what is important here is not that you understand all the details of the experiment. What is important is that these recordings can be used to parameterize kinetic schemes like the one in the big arrow. The kinetic scheme is the representation of the molecular transitions between states. So the molecule is a sodium channel, which means the main ionic channels generating membrane potential changes in the neuron. And this molecule can be represented by Markovian kinetic scheme like this, in which the transition between states is governed by differential equations. The differential equations can be parameterized, extracting the data from experiments like those on the left. So now we have a model that is parameterized. Once it is parameterized, we can reproduce exactly the behavior of the ionic channels, the sodium channels in this case, using the model. So we can transpose the knowledge obtained in biology into a mathematical model. This is reconstruction, okay? Now we go on, and we take the channels, and together with other ionic channels, there are many types, we generate a model of a neuron. What is the fundamental step ahead here? The neuron contains many kinds of ionic channels, as I said to you. The ionic channels are placed in specific compartments of the neuron. For example, this part here that is labeled with an antibody. And that corresponds to here where I'm pointing now, that is called axonal initial segment. This is the point where the ionic channels that I showed previously are placed, not in the other points of the neuron. And this makes a difference. So you have a complex electrogenic system in which the ionic channels are located in a specific part of the neuron. We can do it for any kind of ionic channel. The work takes some time from observations taken in different laboratories, different techniques. Finally, we can reconstruct the whole neuron with all the compartments, and the neuron works as if it was the real neuron. Again, that's a transcription of biology into a model. It's reconstruction. Okay. Now that we have it, we can begin to use it. And the first model that we constructed in the end of the 90s was used to verify the electrogenic hypothesis of this particular cell of the cerebellum that is called the granule cell. The story was that the model predicted the existence of an ionic channel that was not included in the model. So the model was doing pretty well, almost everything except some states of activity were not correctly predicted. And therefore, it anticipated the existence of ionic currents and channels that were not in the model. We looked for them, we put them into the channel, and then we move on into the second paper that came here. So the story began to grow, and here we have inserted all the ionic channels known at that time, and we have predicted some pathologies that derive from that. The point is that here the neuron was able to generate quite nicely the response of the cell, but was not able to predict the amplitude of the action potential, which is the signal emitted by the neuron. The action potential in the model was too big. Why it was too big? It was too big because we needed a different dislocation of ionic channels. We studied it, and we normalized the size of the action potential. That's another paper that came up soon thereafter. Then what happens is that a neuron should be able to transmit the action potentials along the axon, not only to generate them, but to transmit them. There is a law that is called the law of action potential conduction, stating that the action potentials are conducted at constant speed and constant size. They don't decrease in speed, they don't decrease in size. In our model, they decreased in speed and decreased in size. So that was wrong, obviously. So again, we were not happy with our model, improved the model. So we studied the ionic currents in the axon. We developed techniques to do that, imaging techniques that visualize directly the action potentials traveling into the axon. And we corrected the sodium channels. We discovered that the sodium channels are modulated by specific molecules that change their gating properties. We put everything back into the model, and we came up to this other paper here, in which finally we got a model in which the action potentials are generated in the proper place and conducted in the proper way. So that's a long story. I don't think it's finished. Every time we put a hand on the problem, we find something new. And actually, we decided to control the pattern of action potential generation, which means the way they are organized in time, and we discovered that they can be organized in multiple ways, which is not predicted again by our model. And back again, try to understand what happened. So the answer was interesting. By using machine learning algorithms, we discovered that there were not a simple and single arrangement of these ionic channels, but that there were multiple arrangements. And by using these multiple arrangements, we were able to explain, let's say, 70% of the variability. Still, there was one part of the variability that was not explained, and therefore there should have been another ionic channel, yet another. It's a continuous story. In the same year, there was a lot of emphasis on an ionic channel called the TRP that led to the Nobel Prize in 2018. And we decided to look for the TRP channels that were completely unknown in these neurons. They are there, and they explain the residual variability. So again, this is the last story that came out just 1.5 years ago. So basically, I don't think it's finished. If we continue on this line, we will find something new. So basically, these models, the message is, the models based on cellular biophysics are so powerful that, if used properly, can predict the activity in the neuron and its mechanisms, and can lead to discover how the neuron works. So that's the power of this kind of modeling. Then we can propagate up this process into the network. The story becomes very tricky. The network is the most complicated aspect of the central nervous system. The neuron, as you have seen, is complicated but is solvable, is understandable. The network is still very difficult to tackle, very, very difficult. Paradoxically, it's easier to manipulate signals using fMRI on large-scale assemblies than working on a microcircuit. The microcircuit is really the most difficult part to address. And therefore, we decided to build up neural network made of many neurons to understand how this neural network works. Again, Claudia will go much more into the detail. This is the same movie as before, and here you see the activity of some of the neurons in the network that can be tracked and analyzed in detail. This is an example of the story of the network. The story of the network is like this. The network expresses properties that are beyond those, above those of the single neurons, that we call emerging properties, because they are not immediately implicated by the neurons. These emerging properties, for example, are coherent oscillations in the time domain or spatial organization in the spatial domain. And these are the spatial organization and the oscillations predicted by the model. So the model predicts that in certain states, the network oscillates, and in other conditions, the network generates patterns that we call Mexican hat patterns of activity, like this one. Okay, then we went back to the experiments, and using focal confocal laser microscopy, we have been able to discover that there is a very nice Mexican hat organization in the circuit. By using other kind of recordings, not in our laboratory, in other laboratories, the oscillations have been found and demonstrated. So basically, the model, again, is able to predict functional states, this time at a higher level, also for the network. So we're making steps ahead. The most critical point is that there is a theory at the back that was born in the end of the 60s, and is called the motor learning theory. The motor learning theory states that the cerebellum is a circuit capable of performing spatial pattern separation and adaptive filtering. What does it mean? It means that you have a pattern of features, made of features mixed up. The cerebellum can separate these features, group them, like in an artificial neural network. You can work as a classifier, separates the features, and then each one of the features is channeled through a different channel, and in each one of these channels you can tune the feature. So change the delay of the signals, or amplify or de-amplify the signals, filter the signals, whatever. But there was no demonstration of that. An intermediate step was done by colleagues here, also in Bristol, who defined the adaptive filtering theory of the cerebellum. That is a sort of evolution of the spatial pattern separation. They state clearly that the cerebellum works as an adaptive filter, introducing the word adaptive, which means the properties of the filters can be tuned by plasticity, can be changed by learning, as if you have an equalizer and you change the properties of the channels of the equalizer by tuning them. by learning. Okay. That's very nice, but where is the demonstration? Nobody has been able to demonstrate it before. We've done it with the model first, and then we did it with experiments again. And actually, this is the first evidence that the cerebellum works as an adaptive filter. This is the power spectrum of the cerebellar output, and this is the power spectrum after plasticity has been induced. So basically, you see that for each frequency, you have a modulation in space of the signal, and this modulation is changed by plasticity. So this is the first evidence given by the model that the cerebellum works as an adaptive filter, as the theory predicted 50 years ago. The experiments are supporting this, and this is actually something that we published last year again. I stop here, and I give the word to Claudia, because now it's time to use these models that begin to work, now are rather convincing, and to use them in more complicated applications. Quindi io questo te lo stacco, e il tuo è camera.
Sì, aspetta che parto dalla seconda slide, che questa parte da qua.
Vai giù, fai tutto tu.
Okay, so I try to give you some more details and application about this field. So as Egidio said, we want to reconstruct a brain's microcircuit. So we focus on the cerebellum because it's the brain region that on which we work in our lab, but basically the workflow can be applied to any brain region. So first of all, we want to place the neuron with realistic properties into the brain region. So we want to have all the different neuron types. We want to put the right density, the right number of neurons into the volume. And then we apply some connectivity strategies in order to create synapses, of course, between the neurons and be able to generate network dynamics. Then we need to define, let's say this is the structural part of the network, and then we want to put a model of the neuron. So as Egidio said, it can be multi-compartment neuron with many differential equations, or it can be simplified model of the neurons, so like point neurons, maintaining only, let's say, the salient properties of the electroresponsiveness. So with these properties, then we can simulate the dynamics of the network. So we can have the spiking activity, the different dynamics of the different populations embedded into the network. And then if we add plasticity rules, so long-term depression, long-term potentiation on the synapses based on, of course, on experimental data, we can have evolution of these network dynamics, and we can decode a behavior. So we can imagine that a specific neural population, they evolve, they have different response during task repetition, for instance, and this means to have an adaptive behavior. So it can be decoded in motor learning, if we are in motor domain, and we can decode from spikes of the different neuronal population, we can decode a behavior, a learning curve. This means also to embed the network, in this case the cerebellar network, into a sort of closed-loop system. So we have some planned movement, we have some feedback coming from the actual movement, this trigger a learning, adaptation, and then, of course, we can generate this behavior. This is really crucial to understand the neural correlates of behavior, and this is really also, fundamental to understand even brain diseases. So it's the computational modeling in neuropathology can be a powerful tool, complementary to experiment, of course, to help to understand, interpret data, even in motor disorders, for instance. Then, as was already anticipated, this detailed, or let's say, high granularity reconstruction of the network, these networks can be also embedded into whole brain, virtual brain framework, so also connected, in this case, the cerebellar network with other brain regions that can be also modeled with more abstract approach, like neural masses, mean field, something that doesn't take into account the single neurons, but then create a sort of average activity. But in this way, you can really propagate the activity between brain regions, like modeling also long-range connectivity, not only the local connectivity inside the cerebellum, in order to have a wider system. So even to do tasks which are not just cerebellar-driven, but they involve other brain regions. How we reconstruct the network. So we have developed in our lab a flexible code base, a software, which is called Brain Scaffold Builder. It's open source and it's available also on eBrains, which is a human brain project platform. And this can be used to build and simulate different scale of the brain networks. So first of all, we have the reconstruction, which means to define the volume, which can be any shape, which can be an abstract shape like a cube, or it can be really extracted by brain atlases. So we can have really real volumes with folded geometry taken from mouse brain atlas if we are modeling the mouse brain, human brain atlases if we are modeling human brain. Then inside these volumes, we can put the different cell types, so it depends, the differentiation level that we want to keep, so how many different cell types we want, so it can be excitatory, inhibitory, but then we can have much more details and have specific population with different morphology and different electrical properties. Then we need to place these and to connect. The flexibility of this tool that you can really use any placement strategy and connectivity strategy for each neuron type. So it's not really a fixed algorithm that needs to be suitable for all the connection, but you can really be flexible and even introduce a user-defined strategy if you need something more, which is not already available in the tool. It's really easy in Python environment to add your user function and say, okay, for my brain region, I really need this connectivity principle, which was not already implemented in the framework. Then once the reconstruction, the network is generated, so it's a huge matrix with the 3D position of each neuron and a huge matrix of the connectivity. So you have all the connection between the source and target neuron, all the synapses that are present in the networks. Once you have this huge matrix, you can simulate it. And then, of course, we can target different neural simulators because it depends if we are using multi-compartment neurons. So with all the ionic mechanisms, all the ionic currents, of course, there are a certain variables that needs to be monitored, recorded, and used. If you use single point neurons, so just let's say, neuron without any geometry, but just like a point generating spikes, in that case, we have another simulator. I mean, the same network, the same reconstructed network with the location of neurons and synapses, but then we have point neuron models, so it's faster, the simulation, of course, so it's always a trade-off between level of realism, level of details you have, and then the computational load. So if you are doing a very small network, it's fine. If you want to do the full-scale mouse or human brain, of course, we don't have now enough computational power to do a feasible simulation. So you need to leave apart some details and say, okay, maybe some region, I focus on it, I do high granularity model of this, and the other things I do with mean field or just point neuron or something. So you can do a mix, a co-simulation, hybrid simulation in order to be to have a good scientific answer to your question at the same time to be able to run these things on the available infrastructure. And of course, we always use high-performance computing, so parallel computation, big clusters that are available from the Human Brain Project. So there are, in different countries, we have this cluster, and then you can have research on it. So you can launch your simulation on this cluster in order, of course, to parallelize this huge amount of neuron synapse network variables. So this is an example with the cerebellum. So we have 5 main neuronal populations. They are placed in the volume, and they are connected. Then basically we generate a sort of also a ground truth a little bit, because it's a coherent construct of the data, but not everything is available. So something is constrained by the data, so how many source cells converge to another cell type, for instance, but for other connection, we don't have necessarily the synapse densities or other data. So it's like that the model itself, the reconstruction, is able also to predict and to fill the gaps of something which is not already available in the experimental data. Then, as I told you, here we can use a realistic volume. So this is a cerebellar region, flocculus, right and left, so extracted by the atlases. And again, we place the neurons inside this volume, which, of course, they have realistic shapes and a realistic, let's say, amount of voxels inside. So it's a realistic number of neurons. And then this can be done on any brain region, any partition mesh that can be extracted from the atlases. This is an example of the connectivity strategies already available in this framework. So here, for instance, is touch detection. So it's a really high precision way to connect the neurons. So for instance, here we have a red neuron, which is a granule cell. This is the axon, ascending axon and parallel fiber. And then you have the Golgi neuron in blue. And you really search for distance-based detection. And then when the distance is really basically zero, so let's say they touch each other, you define a synapse location. So you connect, and then you can really create a synapse mapping of your network. This is, of course, is also very computational heavy, expensive, because you really search contacts. Of course, you can do optimization of space partition, so you can optimize. You don't really search any combination, but you have a predefined selection of which neurons could be in contact. But anyway, it's really expensive. So there are also other strategy like voxel intersection. So you basically voxelize your morphology of the neurons. So in this case, you have the yellow cell, and then the voxel of the axon, and then the green cell, and you have the voxel of the dendritic tree. And you search which voxel intersects, and then you say, okay, when inside the intersecting voxel, I can create a certain number of synapses. It's a little bit less expensive in terms of computational load for the algorithm, and then you can place all your synapses. Then you can simulate your network. As Egidio presented, this is an example of the cerebellar cortex of a mouse. Here you have the mossy fibers coming from outside the cerebellum. Then we use a stimulation pattern taken from experiments. So it's like spike trains on this fiber, which are associated to like a whisker path on the mouse. And then to see how the propagation comes into the network. So it's a background on all these fibers, which is the in vivo state, so they are not silent, completely silent. And then at some point, a whisker facial sensory stimulus is provided, which correspond to a very strong burst of spikes lasting 20 milliseconds on this bundle of fibers. And then we can monitor the activity of all the cells potentially, of course. It's a huge amount. It's also a big file of results when you record this kind of simulation. And then here you have the voltage membrane of some granule cell, Golgi cell, Purkinje, stellate, and basket cells. All the neuron types you have in your microcircuit. And then, of course, it's super easy to manipulate some parameters and simulate some different states. So we can say, okay, let's do a different release probability at the synapses. So it's like to simulate plasticity states, long-term depression or long-term potentiation, and then you see the completely different response of the neurons inside the network because you change the release probability at the input of this network. So you change the propagation and the level of activity. And these are some videos of this. This is a single neuron inside the network, and then with the different synaptic currents, and here it's the just point neuron networks, the equivalent, just plotting only the somata of the neurons, not the whole morphologies, and this is the activity with the stimulus. So, of course, once you have the validation, this is powerful to predict and monitor the signal propagation and the spatio-temporal processing of the signals. This can be done with single-point neurons, so we have only spiking patterns, so we don't monitor all the synaptic currents because we don't have these details, but we can monitor the raster plots, let's say, so the spiking activity of each neuron into the network. And in this way, we can put some plasticity rule, as I told you. So there are some connection between population that are plastic. So the synaptic weights continuously change based on errors that comes or other rules that comes into the network. So here we have some plasticity rules. So just to understand, for instance, between the granule cell and the Purkinje, we have specific kernel, and then the synaptic strength is modified based on the timing of the pre- and post-synaptic spikes. So depending if they are correlated, if one is responsible for the other, if they fire together, or if they are uncorrelated, the synaptic strength is proportionally modulated. And then there are different synaptic sites, so it's a distributed plasticity. So actually the cerebellum has more than 20 forms of plasticity. Not everything is in the model, but there are different plasticity rules with different time constant, which means to have fast adaptation, consolidation, memory, or recalling when you do test-retest tasks. So all these mechanisms, they are a little bit partially modeled into these learning rules. So we simulated a very simple task, so eye blink classical conditioning, because it's a cerebellum-driven task, where you have a conditioned stimulus, like a light, and after, let's say, 300 milliseconds, you have an unconditioned stimulus, which is an air puff. What's happening is that at the beginning, you have the blink after the air puff, but after some repetition of this pattern of stimuli, the system learns to anticipate the unconditioned stimulus, so you have a blink which anticipates the unconditioned. So the eyelid is anticipated, and this is driven by the cerebellum. So, for instance, the patients with cerebellar damages, they are not able to learn this association, so they don't produce conditioned response, which is the anticipated blink. Or they learn in a very long time. I mean, this is also use a biomarker for disease at the cerebellar level. So we have neural recording from the mouse during this task, so we know how some cells, the Purkinje cell, behave. So at the beginning, they fire in this way. After a certain number of repetition of the task, we have different spike patterns. And then the deep cerebellar nuclei, they are inhibited by Purkinje cell. They have, of course, let's say, opposite firing. So when the Purkinje cell doesn't fire, this means to release the deep cerebellar nuclei cell to fire. And this triggers the conditioned response, the blink, which anticipates the unconditioned stimulus. So this is driven by this mechanism. And then so this is the condition. You see at the beginning, the blink is just after the violet line, which is the unconditioned stimulus onset, while at the end of learning, you have an anticipation of this. So this is a recording from other collaboration on behaving mice with electrodes implanted, of course, during behavior. And then this is the curve, so you can see the learning of condition response. And when you have cerebellar groups, so with very severe lesion of the cerebellum, basically they don't learn any condition response along trials. Okay. So we reproduce with our network, so we know where the input goes, so mossy fiber, climbing fiber, then we have all the dynamics inside the network, all the neurons, all the plasticity, and then we can monitor the spikes of the population at the beginning of the learning, at the end of learning, and then we can decode the behavior. So here we can see the different modulation of the spikes, of the dynamics of the neurons. We can see what increase, what decrease, and then we can, generate the curve of the conditional response. So this is a way to reproduce the neural correlates of behaviors in model, and then we can manipulate this model and create an alteration, reproduce in pathology, and see the impacts on behavior. Then we use this model in other tasks. So we use to simulate saccadic movement, where the cerebellum play a crucial role, again with the models, and we can see the evolution. So from the first trial till the end, we have some increasing of activity, decreasing of activity, so burst pausing, all these network dynamics that are able to tune and generate the motor responses in the precise timing, because also, of course, is really important that this increase of the output of the cerebellum anticipate of a certain time the unconditioned stimulus, so the air puff, not any time. So it's a time-locked motor response due to the stimuli association. Then we use with also the neurorobotic platforms to simulate a whisker system driven by the cerebellum. And this is what I told you before. So we put the spiking cerebellum into bigger controllers. So we have a planner, we have a sort of M1, so motor cortex, rough controller, and then the cerebellum help to compensate for perturbation from the environment, or depends on the task. But it's a controller that includes other things, not only the cerebellum, of course. Then, as GDSA, we basically don't have a peripheral system. So from the brain, or the central nervous system in general, we interface directly with a sort of plant, so with the body acting into an environment. So we, at some point, we will do embodiment and we'll add spinal cord modeling and these things in order to have more realistic decoding from central nervous system till the actuation of the body. So as I told you, we are doing different works in pathology. So we introduce some alteration in the single cell, and then we try to propagate this alteration of a very low-level mechanism into the network till the decoding on behavior. So we want to predict the impact of specific lesion, and we are working on autism, on dyskinesia, on ataxia, where we know that maybe a specific ionic channel has a different conductance, but they don't know exactly how this impacts at other levels. So we introduce this alteration into the model, and then we simulate the whole flow. Okay, with the final goal to have neural correlates of behavior. And then the last thing I will show you is some works that we did in putting this kind of brain models in the controller of real robots, so not just, let's say, in a simulation environment. Of course, this means to simplify some parts because we need the real time, which we don't care usually during simulation, especially if we have multiple details with all the ionic mechanisms, the real time is really far. But if we want to control something to have an adaptive behavior of a robot, of course, we need to have a models able to go in real time. So this is an example done with Polytechnical, with a very simple, let's say, robot, so a now robot. So we have tracking task, so it needs to do a circular task. And then inside here in the software controller, we have a spiking cerebellar network with the plasticity. So basically what I showed you before with some simplification in the neural populations. And then at some point, we introduce a perturbation, so now, and then it's like a weight, or anyway, a perturbation, and then, of course, it starts to have a big error due to this unpredictable event, because basically the cerebellum had learned an internal model, so it was in a predictive way, it was applying a model that was learned, but some point, thanks to the error and to the plasticity mechanism side, it changed the spiking activity and then, of course, also the output decoded from the spikes. So this changed, and then when here it's at the end, so basically back to the right trajectory, and then at the extinction phase, so suddenly the perturbation is taken off, and you have the so-called after effects, because you compensate something that is not there anymore, but you expected that. So then for few trials, you still do an overcompensation, but then again, thanks to the plasticity, which are even faster in the extinction, you can go back to the right trajectory, let's say. So this was an example of a realistic cerebellar model spiking in real time, controlling some real behavior with all the, of course, inaccuracy of the real robot and the interaction between body and environment. And this is in the direction, of course, of having explainable brain-inspired artificial intelligence into robots, of course, just a small step. Okay, so this is a big work from University of Pavia, Neurophysiology Lab, Neurocomputation Lab, brain modeling, and then a lot of collaboration from all over the world. Okay, I think, I don't know, Egidio, you...
No, it depends on whether you survive another 2 minutes or not. What should we stop here? Probably we better stop here.
Maybe some questions. Well, thanks a lot. Is there any question from the floor?
You mentioned neuroplasticity, and I want to ask the reconstruction of the synapse. Have you considered about the ASTROCYTS?
Consider what, sorry? Maybe I don't know what is it.
The second question.
Of neurals, astrocytes, and microglia. This? Yeah. Okay, Egidio, this is yours.
No, we didn't consider, I mean, the rules that we're applying here are extracted from a large set of biological experiments, and they're abstract rules. So we are not implementing the mechanism of plasticity. We are implementing only the effect that certain patterns have on the plastic process. Therefore, we disregard the fact that they might originate from the intervention of microglia, glia, or whatever. It is not written into the model. We have a phenomenological description of the plastic rule. Okay. However, if I have to tell you about plasticity in the cerebellum in terms of mechanisms, there are about 20 forms of plasticity with very differentiated mechanisms at different synapses in the circuit, but none of them really involves the glial cells, because nobody studied it, probably. Yeah, because if somebody starts to study the impact of glial cells on plasticity, surely find something. You know, biology is something like that. Yeah.
funzioni. Cost function.
Lost function,
I think we
Claudia?
called cost function. Maybe.
Cost function.
But maybe it's the same
Cost,
thing.
or
It just
cost function, you mean? Well, the cost function is something that the principle of the cost function is embedded in the principle of addressing the distance between a predicted trajectory and an effective trajectory. So, for example, in the robot, when you saw the red line and the blue line, no? The distance between these two lines addresses the concept of the cost function. Minimizing the distance means minimizing the cost function. So the robot is minimizing the cost function, basically. Through what? Not using an algorithm, an explicit algorithm, using an implicit computation based on the feedback that goes into the circuit. So the error goes back into the circuit, triggers plasticity. Plasticity works in a way to minimize the cost function. So this is the great intuition of David Marr, who did it in 1969. He had this incredible intuition without knowing anything about the mathematics of the cost function, but he set out the principle more than 50 years ago. That's incredible. So we're beginning to understand what he did now. But that's exactly what he say, actually. Also. Yeah.
Any other questions?
Yeah, please.
Since you mentioned Marr, computer vision people might be here, and so I guess there was always these different levels, and you mentioned a lot about bottom up and top down for integrating. Do you think there's he sort of also was proposing that we studied in the middle of it as well. Do you think that there's people starting to do that? I guess there's been a lot of work on both ends, but unifying them still seems elusive. We're seeing people doing more of that.
That's a great question. Can I go?
Oh, yeah.
Actually, Marr had a global vision of the function of the brain and used the statistics of circuit activity, he didn't know very much about activity. He knew about the statistics of connectivity, which is even more difficult for me to understand how he could get to that intuition. But he realized that the statistical organization of the circuit was good enough to explain a global function, okay? So he didn't put anything in between. He had this statistical organization of the network. He had behavior on top of it. And then he introduced a fundamental concept, plasticity. He said we need one plastic synapse. In his idea was the one between the parallel fibers and the Purkinje fibers, which actually applies, is true. And he thought that there was an error running in the system, and this error was coming back to the the Purkinje neurons through the climbing fibers. So by setting this out, he had this great intuition of how the whole system works. Actually, in the real system, we had a lot of steps in between. And one first step is the way the neurons work, no? And this we consider. So now we have the neurons. He didn't have the neurons, now we have the neurons. The second step is the precise organization of the network, because in a statistical description, he lost all the geometry. So the geometry was lost. It is funny because the cerebellum is the most geometrically organized structure in the brain, and he knew it very well. He knew it perfectly. But for the sake of simplicity, he decided to forget about geometry. Okay, so now we have geometry, so neurons, with all the nonlinearities in neuronal computation, geometry. The third point is that he said we have plasticity, a global concept of plasticity with a specific location. Now we have more than 20 forms of plasticity, not one. And now we're considering many of them, not yet all of them, but many of them. Then there is another layer of complexity in the middle that is the specific connectivity of the cerebellum. Because as Claudia said, we are connecting the brain to the robot. But between the brain and the robot, we have the brainstem, the spinal cord, the muscles, all these kind of things we do not have for the moment in the model. That is something that we discussed this morning. It would be very important to have it. And I'm pretty sure that the story will become, on the one side, more complicated, on the other side, much more understandable for everybody.
Follow on. Now everyone will hear me. I suppose it's a follow-on question. So you just hinted that your model at the moment does not include the spinal cord, the peripheral nervous system, the end effectors and the sensors. And which of those do you think is the most vital, and what do you see the impact that they would have? And I can see the effect of the spinal cord and the muscles being very important, but the peripheral nerves themselves outside of the spinal cord, there are no synapses. It's a fairly straightforward transmission system. So what do you see as being the important features of those different systems, and what impact might they have?
No, yeah. I see it.
No, listen. The cerebellum does not contact directly the spinal cord. It has to pass through either the cerebral cortex in the so-called forward loop, or in the inverse loop goes to the spine, to the brainstem, and then down to the spinal cord. So it anyway doesn't contact ever the spinal cord directly, but passes through other structures. So if we misregard the other structures, we make a mistake, in principle. For the sake of simplicity, we can begin to do without them, but then we have to put them. So we are at this point, so we need to put them. I think to, let's say, make an hypothesis, a constructive hypothesis on what to do, we have to consider that motor control eventually depends on two kinds of motor neurons, the alpha and the gamma motor neurons. And the gamma motor neurons controls the muscle spindles. The alpha motor neurons control the muscle fibers. The cerebellum goes on the brainstem, activating basically three nuclei, descending nuclei. The red nucleus, the reticular descending nucleus, and the vestibular nucleus. The vestibular goes on the alpha motor neurons. The other two nuclei go mostly on gamma motor neurons. So without considering this element, we are missing a lot about what the cerebellum is doing. So this is clearly what I would like to have into my system. When you go to the spinal cord, but there you have another word to consider, because the spinal cord is made of circuits, it's not made of neurons. These neurons are connected into circuits, and these circuits are good to control the reflexes. In this model, we do not have the reflexes. Without reflexes, it's like I did an example this morning, it's a bit, I would say, crude, but it's like trying to drive a car without the springs and, what's the name of ammortizzatore? Damper, with the dampers. So you will never be able to control the dynamics of a car in a circuit without the dampers. It will go out of road. At the first turn, it goes out. So the point is that this is what we are trying to do. We control the body without the dampers. We need them. So this is another crucial element we have to consider. And the gamma system controls the dampers. So we have an intricate mechanism of controlling movement that we are completely misregarding. At the point where we are, because, for example, you showed the eye blink classical conditioning, we don't need the dampers. We only need something that moves up or down. It's like a digital control. And in the digital control, you can run without any precise representation of the body, of the effector, of the actuator. You just put the actuator and you actuate it. That's it. If you need a complex system like arms, legs, parts of the body that move dynamically, and then you need all these other elements. So I'm pretty sure I'm a strong supporter of this idea of including more components in our system. But this takes time. So we can't do it just in a rush. Yeah.
Please.
You say the neural system as a sequence or digital system, but how do you think with the memory function? Is it continuous or discrete? If you think that as a continuous, what's the perspective of brain itself to think the continuous things?
Yeah, probably your question is more, can be rephrased into learning, if learning is continuous or discontinuous. Yeah, because memory can stay there for some time or forever, but that's a question of duration, not of continuity. So learning can be continuous or discontinuous. Yes, that's true. Well, that's an important point. You know the problem of destructive interference, no? If you learn when you act, you are at risk of destroying what you have learned before. So there must be a sort of mechanism to control when you learn and when you do not learn. Our understanding of this mechanism is very poor, I have to say. So what I can tell you is that the cerebellum learns continuously because when signals flow into the cerebellum, they are continuously compared with a template that is coming from the commands. It's a kind of supervised learning in which you have a template that is the command issued by the cerebral cortex, and you have a result. You compare the result with the template. And this is a continuous process. It never stops. It never stops. So in my understanding, the cerebellum works in continuous learning mode, in my understanding. I may be wrong. I may be wrong. So at least for the task that I am able to imagine, to envisage, is in continuous learning mode, but I may be wrong. That's an important issue. Okay. Sooner or later we have to consider this as well. Yeah. Well, all the system learns, the learning function is a continuous function in time. And there are learning functions on all the kinds of synapses that have plasticity. For example, between the mossy fibers and the granule cells, there are several forms of plasticity, not yet in the model. Between the parallel fibers and the Purkinje cells, there is plasticity. It is in the model. Between the Purkinje cell and the deep cerebellar nuclei, there is plasticity. It is in the model. So we have already some plasticities in the model. These learning rules are continuous function of time. So when there are signals running into the learning function, I would say, these learning functions start to work. They generate an output.
But I think if converted to the output, it will come through the single neurons, active or inactive, as we can say.
If the neuron is inactive, nothing happens. If the neuron is active, something happens. I hope I understood properly your point. Maybe
I got a quick question for Claudia then, please.
No.
So I saw that you're using actually a big model with different micro models being multi-scale, I guess. How do you deal with the interaction between those one and the potential error cascade that you have?
You mean different from the cerebellum? So when we put in the system
Yes.
with planar or something. Okay, now we have different, we are working with other groups, and some other groups, they are building spiking models of the other blocks. Let's say that for a first system, the other blocks are like analog blocks, so they generate signals, but not really like neural population with specific connectivity. So it's more like an input signal that is converged on the input of the cerebellum, which are the mossy fibers, but it's not like really a one-to-one synapse connection like inside the cerebellum. So now the other blocks are more abstract based. So like the M1 generates a rough inverse controller to have a motor command. And then they are put as spikes. So we have always interface between, let's say, a block to transform spikes into analog and analog into spikes, so decoding strategy. And then this comes into the cerebellum, then is compared to the actual trajectory, learning, and so on. At some point, when also we will have, with the other collaborators, a complete spiking model of the cerebellar cortex, for instance, of course, we will have specific population directly spiking inside the cerebellum. So it's more like very one-to-one connectivity with synapses, maybe plastic even between different brain regions, and then it will be a whole brain. But now some blocks are really abstract blocks. So even the state estimator, which is probably ascribed to the parietal cortex, so you have the prediction for the cerebellum, the actual feedback, and then it's compared to define the state of the body. And this is, let's say, it's a spike-based operation, but they are really abstract. You don't have neurons that with specific dynamics that they do that. So it's still a system to be completed somehow.
Thank you. Right. I think we reached like a good time to move out. So I would like to thank you, the speaker, and see you to the next seminar.